Okay, let me just give a 10-second introduction again. My name is Shivika, and I'm a youth ambassador for the Aneurysm and AVM Foundation. Super excited to be here today telling you about the scientific research I've been doing, and I promise I'm going to try to make it like the least boring as I possibly can. Hopefully, this is exciting for everybody. Okay, so first of all, why did I start doing this, and why am I even here today? So lost my grandmother when I was 14 years old to the rupture of an undiagnosed brain aneurysm. So basically, she had a brain aneurysm in her brain. It ruptured, and we never even knew it was there. There were no brain scans done prior to show that she had a brain aneurysm. But I'm sure, as you all know, when it ruptures, then the whole vomiting, seizing, her eyes flip back in her head. She completely lost consciousness. She went into a coma for about a week, and then that eventually led to organ failure and her passing. So my grandma and I, we were always super duper close. We were actually kind of like, I think she was my best friend for quite a while. Actually, I think throughout my entire life, I think she would have been the number one person I would turn to talk to for anything. So obviously, I was very devastated by this, and even today, it really, really affects me. But I think one of the main questions that I first had when my parents told me that she had a brain aneurysm was one, what is a brain aneurysm? And two, how do we not know it was there? When it ruptured, of course we knew. I mean, she was vomiting, she was seizing. This is actually a picture of one of her scans where I'm not sure if you can see, but the little line in the middle of her brain that's supposed to separate the two halves evenly, she had such a big rupture that it shifted and it bent. So completely transformed her, and obviously she wasn't able to survive that. So how do we not know it was there? So then I started going to my trusty dusty research assistant, Google. I literally sat down on my grandparents' bed in India at like late at night, Googling things about brain aneurysms about 5 months after her passing. And that's actually how my research started. So just to take you through a little bit of my research journey, I've always been interested in early detection because I know that there are different surgeries that you can do if you know that you have a brain aneurysm or an AVM or any kind of condition that's similar to that relating to the blood vessels in your brain. However, if you don't know that it's there, then you can't take any action. And especially for brain aneurysms, they don't present as any significant symptoms. The max someone would get is a headache. And of course, they would likely think that they would not likely attribute that to having a brain aneurysm. So, how do you find out what's going on in your body beforehand? So, right now, the thing is that most people use imaging, MRIs, CT scans, CTAs, but those are so expensive. There are so many zeros to the back of that one digit that I can't even count. And it gets very hefty bills, and it's really, really hard to monitor that over time. I don't think anybody would go to their doctor on a regular visit and be like, hey, I need a brain scan today. I don't think that that would really work out. And it has not, unfortunately. So, what I was thinking was, what's something that we do routinely? Blood tests. So I started searching up different biomarkers. So biomarkers essentially are molecules and proteins in the body that basically tell someone what's going on without having a need for imaging. So I, through my various amounts of Google searching, I ended up writing and publishing a research paper on a variety of these. And for me, so some of them were inflammatory, so they lead to the inflammation of the vessels and then weaken them. Some of them are genetic. Some of them are multiomics, which is just a fancy word for saying it's a bunch of interactions between proteins and DNA and a bunch of all that stuff. But my most interesting find, I would say, was blood-based biomarkers. So these were actually the things that I was thinking of, as in we could create kind of like a holistic panel that a doctor could order for someone to get these molecules tested in a blood test. So from your blood, based on certain different markers, you can find out if you are at risk for a brain aneurysm or not. And just a couple of my results, I wanted to really highlight how significant the differences are. So these biomarkers are present in everybody, but in patients with brain aneurysms, they are significantly higher. For example, these are just some figures from my paper. If you can see here, the red dots are the ones for aneurysms, and the blue dots are patients who didn't have an aneurysm. These are, by the way, this is just a compilation of different studies and research that was done. So essentially, if you can see, there's usually quite a significant difference between the levels of these, in this case, it's genes, the levels of these genes for aneurysm patients versus normal patients. And on this side, for some reason, the eighth grade me switched up the colors because red totally means control and blue means aneurysm, apparently. So I switched these up. But as you can see, for patients with a brain aneurysm, again, this molecule is much, much higher than patients who don't have one. So that's essentially the concept that I based off my initial research. Now I moved on to how can I get like instant results? So then the following year, or actually no, I think 2 years afterwards, it took 2 years, I coded an app to flag for the risk or again detect brain aneurysms from CT scans because CT scans are often about half the cost of an MRI. Now that was great. I coded a certain like score that was able to help me minimize false negatives because false negatives essentially in a real world setting would mean that someone has a brain aneurysm but it goes undiagnosed, essentially leading us back to square one. So that's of no use and of course it's very, very dangerous. So in the end, for that recall score, the closest it can get, I guess the way you figure out if your app or your model works well is if it has a score that's closer, it gets closer and closer to 100%. And what I coded was one that had a 92.1% recall score. So I was really, really happy about that. But then I kind of thought back to the accessibility problem of imaging. And again, imaging, you can't monitor it over time. You can't go take a scan every day, see if you have a brain aneurysm or not. So that is actually what led me and a friend that I worked with to develop NeuroAcoustix. So NeuroAcoustix is a device which essentially listens to how blood flow sounds in the brain and uses that to flag for the risk of a brain aneurysm. So to simplify that a little bit, if we think about a pipe, you have a pipe and you have water flowing through it. It's going to create a whooshing sound, right? But if there's some kind of a bulge or, I don't know, some kink in the pipe, the type of sound that you hear is going to change. And that simple concept is actually what we base this off of. If you imagine the pipe as your blood vessel, if there's a normal blood vessel, a healthy one, and blood flows through it, it's going to create some small vibrations and some small sound signatures that we can capture and record. But if an aneurysm forms, then that sound is going to change. So essentially, our hypothesis was that blood flowing through the circle of Willis, essentially the main network of arteries in the brain, that's going to the blood flowing through that is going to generate the small sounds that we can use and analyze to flag for the risk of a brain aneurysm. So to go into this a little bit further, this is essentially how we envision our device because the device still is in its early stages being used in a human. So essentially we have these two small mics. They're placed above over on the eyes, which I'll get into a little bit later, but they're placed on the eyes and they record the sound of blood flowing through your brain or the blood vessels in your brain. And then we connected it to a small computer, which already has a bunch of different code loaded onto it. And eventually it just outputs whether someone's at low risk for a brain aneurysm or high risk for a brain aneurysm. So with this technology, what someone can do is, it's honestly like an early monitoring system. That's how we designed this device to work. So if someone, let's just say, takes a reading every 1 or 2 days and the device, God forbid, outputs high risk, then they can take this to their doctor, order some more testing, and then hopefully catch a brain aneurysm earlier. So the process that we use to do this, obviously we don't have access to patient data. We're both minors, so we couldn't run any clinical trials. So what we did was we tried to innovate and get as close to a patient blood vessel as possible. And I think that's one of the really interesting portions of this research. So first thing we did was we used CFD, computational fluid dynamics, big words. Essentially, we just used math to model how a fluid would flow through the circle of Willis. This image on the left here is actually of an MRI from a patient that some researchers at the University of Michigan published. They actually used that, took away all the brain tissue, and just left the circle of Willis. we put that into a software where we flowed artificially, like simulated blood flow through this circle of Willis, and we recorded the sound response to blood flow. We had to use some kind of specialized equation, but that's just really, really complicated. So I'll not go into that right now. But that equation, eventually, the results showed us that there is a clear difference between the sound responses for these circle of Willises that did not have a brain aneurysm and also those that did. So that, I mean, we just conceptualized this off of literally a pipe with water. So we got really, really excited by this because we found that our simple concept was valid in a mathematical model. So that was actually our first step. But of course, this is all theory. We wanted to test it a little bit further. So then we did a little bit of mechanical testing. So we had our models, the ones that we used for our math stuff, and we just decided to 3D print them. We had some with aneurysms and without aneurysms. So what we did was if you see this little red network, those are our 3D printed circle of Willis models. This one, it does look like this one is one with an aneurysm. I think it's about right here. They're actually very, very tiny compared to the whole network of blood vessels. Then what we did was we embedded them in a special kind of jelly because that jelly actually mimics how brain tissue is, or the density and elasticity and properties of that brain tissue. So again, just trying to get as close to a patient brain as possible. And then through this little pipe, we flowed artificial blood through this while our device, which at that point was in a very, very bare bones stage, it recorded those sound responses and analyzed them. And these two they almost look like little sticky notes or pieces of paper. Those are actually our microphones. Those are our little sensors. Those capture the sound that we're looking for. And it's honestly really, really amazing how something that thin and fragile could record sound responses. It's really cool. And then we moved on to biological testing. Now, this was a little bit more complicated, but I think this is one of my favorite parts. So again, mechanical testing is really cool, but obviously we used 3D printed models. And although we used some kind of bendy material to print it out, it's obviously not as close to a human blood vessel as possible. But you know what is? Something else in biology, leaves. So we took spinach leaves, and spinach leaves, if you know, they have little vein structures in the middle. And they're branching, and that honestly is actually quite similar to how blood vessels are in your brain. They have a branching structure. So we chose our spinach leaf, and essentially what this, again, way too complex, way too complicated diagram is showing is that we essentially stripped the spinach leaves of its plant cells. So that actually left behind just the vein structure and the rest of the leaf, but it had no plant cells left in it. Then what we did was we took human aortic endothelial cells, so essentially the cells that line blood vessels, and we put them into this little corner right here so that they could line the inside of the vein structure. And once they started growing there, they almost formed like a little blood vessel in themselves. Then what we did was we injected our artificial blood through there while our microphone again recorded responses. So this is essentially how we tried to make our models as close to a human brain as possible. And for our mechanical, this one, the 3D printed one, and our math models, we found that there's, again, a clearly measurable and very different sound response that we get when we flow blood through vessels with aneurysms and vessels without. So we were super duper excited about this research because it has so many different possibilities. We're hoping that we could develop this further because right now it's kind of an early screening mechanism, but maybe if we start doing some human trials, we could turn this into something that's almost like a short diagnosis or something that's at least a lot more accurate so that we can give this results to a doctor, or maybe a doctor can prescribe a patient who's at high risk with this. We don't really know, but we're really, really excited about this. So yeah, I think that's my research so far. And this I actually took to a huge science comp, and it was really, really fun meeting everybody, getting to talk about my research, learning a lot of new perspectives, actually. But it was just genuinely an enlightening experience. And for me, I felt like it was a lot more personal because it was a really nice tribute to my grandma. And she's not here with me today, but I'm going to try to do as much as I can in the most nerdy way possible to make sure that other people get the chance that she never got. And I would absolutely, I know that we here are a group of survivors and people who are supporting others. So if any of you have any feedback for me, my research, I feel like it's useless unless I can help it get to the people who are going to benefit from it the most. So if you have any ideas on how I can improve this, or if you have anything you absolutely want to say about this, please let me know. I am so happy to take your feedback and I am so excited. And thank you so much for your time.
Woohoo! Very well done. I know you are presenting, so you're not keeping up with the chat,
Oh my gosh.
but you're
Ah, 20
getting
messages.
lots and lots and lots of love in that space.
Thank
Just
you.
an amazing job, Shivika. Thank you for sharing it with me. And yeah, we definitely will be taking some questions. So we'll start with you, Vinny. You've got your hand up.
Yeah, for oh my God, Shavika. Wow. First of
Hi.
all, like, come on, round of applause. That was amazing.
Thank you so much.
Like, can you help put this in perspective? How old are you? Like what
I'm
what grade?
17.
Okay,
I'm
okay. So
17.
because like this is like a capstone. Like this is like you're getting every scholarship I've ever heard of. But yeah, amazing like design and your hypothesis is amazing just in
Thank
general
you.
because as soon as you said it, I was like, oh, duh, of course that makes sense. Like, and it instantly made me think of like an echocardiogram that,
Yeah.
and like an ultrasound, like when you're looking at a baby through the stomach and you can hear the whooshing sounds
Hmm.
of the blood. So like it just it's a hypothesis that I get like it's so profound and yet it's such a good design and everything and your models are really cool. Did you ever think of so I know that you use decellularization using these spinach leaves as you
Hmm.
discussed. Did you ever toss around the idea of using some sort of like other animal vessel? Like I know pigs are often used in medical research, like some sort of pig vessels that you could
Yeah. Hmm.
did you ever
Absolutely.
consult
So
about that?
yeah, absolutely. So I think one of our next steps for this project, we are not stopping here, don't worry, is
Yeah.
we're going to be taking this into animal research. We
Yep.
started off with this because animal research requires a lot of different permissions, and neither of us, we don't have a mentor. So we
Mm,
would need an adult supervisor and someone who could guide us through the process of animal research. But once we hopefully, I don't know, find a mentor, someone who could help us really take this further, we were thinking of pig vessels, mouse vessels, especially because mice are so similar to humans. But yeah,
right. Yeah. But even just, the idea to use decellularization and then replacing it with human cells is just brilliant.
thank you.
Um, what kind of models were you using in your like machine learning models? I
Hmm.
mean, because I have a background in computer science, so I'm
Ah,
somewhat familiar
awesome.
with the stuff you're
Yeah.
talking about.
So for our machine learning, we use two classification models. They're called random
Mm-hmm.
forest and logistic regression, just because we
Yep.
had a small limited data availability. And we optimized for recall once again, just to minimize false negatives. Those were our
Yeah,
goals.
very familiar with classic conditioning and all that stuff. So we can nerd out about that later. But
Oh, sure. I love
anyway,
that.
amazing, amazing, amazing.
Thank you. Thank you so much.
Once
I appreciate
again,
that.
so cool. I'll
Thank
let someone
you.
else have the floor now. Bye.
Stephanie.
Hi, Shivika. I'll try
Hi.
not to cry because you know how proud of you I am and
Oh,
so
thank
happy.
you.
And I don't want you to brush over the science fair that you went to. I would love for you
Sure.
to talk about that and how
Yeah,
huge it was. And if you would, that would be amazing.
absolutely. So the science fair that I went to, I didn't get to go into too much, but it's called the International Science and Engineering Fair, or ISEF for short. And essentially, the way it works is that you compete at your local county level. For me, it was county level because our school doesn't do stuff like this, but county level fair. And then some projects are selected to go to either the state level or the international level directly. I was qualified directly to the international level. So I got to compete and take this research with my partner to a forum that had some of the brightest minds from around the world. I mean, it was really a privilege to be there and see what everybody's working on. Let me tell you, it's amazing what the things we can do. And just really, really excited by that science. But I had a wonderful time. It was 1 week long. And actually, oh my gosh, I did win a special award there. So it was a really rewarding experience. And it was just, I feel like it was amazing to do for my grandmother's memory. I mean, for me, I feel like this research, it matters to me a lot more than the awards and the conferences. It matters to me because it's a tribute and it's very personal to me. And I think it's really funny because my parents, oh my goodness, I know some relatives of mine, not my parents directly, they were very supportive, but some relatives of mine called me up and were just like, hey, why don't you do some research on some other disease that might get, I don't know, some more awards? And I'm like, no. And we start having arguments because I want to work with this disease specifically because of my personal connection to it and losing my grandmother. So, yeah, I mean, it's kind of being a little, I mean, this disease, it's definitely not researched enough. I believe only $2.47 are allocated per person affected by the federal government, which is paltry. So hopefully we can change that in the future. With all of you advocating and even being here for yourselves and your loved ones today.
Thank you.
Rochelle, you are next.
Yeah, I'm sorry to hear about your loss, and
Thank you. I
I
appreciate
don't
it.
want to minimize it with this comment about
Not at all.
myself because that bugs me. But I just wanted you to have this to add. I'm a burst pica aneurysm survivor, and I
Bless
went
you.
into a coma and had a C1 laminectomy
Mm-hmm.
and had a VP shunt put in. So it was a lot, but I never got the thunderclap headache.
Hmm.
And what I had, I was on the phone speaking to my sister-in-law, and I said, that feels weird. And then I thought, maybe I'm going to call 911. So what I felt was, I felt a pop in my head. And then I could hear popcorn popping in my head and feel it, which
Hmm.
was kind of weird. That was the
Yeah.
sensation. The neurosurgeon said later that he felt it was the blood bursting out of the vein against my skull. So I never had that headache. So I called 911 pretty much immediately. But as I called them and spoke to them, I could feel life draining. I knew I was going to lose consciousness. And I told them, and I did right at the moment. I said, I think I'm going to lose consciousness now. And then it happened. But just for your study, I wanted you to know that some people don't realize that, you look for the thunderclap headache, but I never got it. And it can be other things. And for me, it was this popping, which isn't unusual, I found, but people always talk about the headache.
Mm-hmm.
So I
Yeah,
just
that's
wanted to add that, and I think you're wonderful.
oh, thank you. I think you're wonderful too.
Thank
But
you.
and I
Shivika,
think
I've
this,
got a
yeah.
question for you.
Oh, yes, please.
Okay, so as I've expressed to you before, fluid dynamics in terms of brain aneurysm research is dime a dozen. But the reason it's dime a dozen is because it is such an obvious place to be in that everyone is trying to find something unique about it. And I do believe that it took a 17-year-old brain to come up with something unique that adults are not doing.
Yeah.
Um, what I would, I don't even know if this is a question. It is a question, but you may not have even close to the answer because you're in the midst of still studying. My question is, how did you decide what a quote unquote normal sound was? And what would make an abnormal sound when there are so many other things inside of our vessels, like, for example, a clot, right? A
Mhm.
different kind of stroke that would cause it to be abnormal. Where are you at with normal versus abnormal?
Yeah, so currently we're at the normal. So the way that we did this was we classified our aneurysm sounds versus our normal sounds based off of the data we got from our mathematical models because those seem to be the most isolated and accurate. So we set the threshold. We actually set the threshold a little lower than we would normally do for to classify for an aneurysm sound, just to make sure that we're getting as many cases, or not as many cases as possible, again, minimizing the risk of missing an aneurysm case. Now, currently in that whole stage, we are still at just an aneurysm sound versus non-aneurysm sound at right now. But again, as I think one of the main things that we're looking for is more patient data, because as soon as we start recording this on patients, we're going to revamp this entire thing. Let me be honest with you. We're going to revamp the whole thing because of how much stuff is in this little space up here. We have our skull, we have fluid, we have brain tissue, we have other vessels, we can have clots, we can have, for people who have had previous aneurysms, clips and coils, things like those. So it's going to change absolutely and completely. And obviously, we have to take into account comorbidities, other diseases that could affect all this. So long, long way to go. This is just the starting, and we are definitely aware of how much we still have to learn and so much we still have to explore with this. And we will be definitely studying that more and taking that into account.
I really appreciate your answer. Uh, Anna, you are next, right? Yes. Miss Blythe, can you hear me now? Hmm. Okay. I will come back to Anna. Vinny again.
Yeah, sorry, just what you were talking to Dina about, you know, like I said, background in computer science. I know conditioning and I know, um, networks that find a, what's the word? Find a label for something. But yeah, it seems that experimentally you could somehow collect pre-labeled data um, as you're training the model, like in order to train your model and train it on different sounds. So that part doesn't worry me at all because I think you'll have more than enough of that if you get funded for real human trial research. Because yeah, essentially, uh, you know, Dina, what would happen is you know somebody has an aneurysm already, and then you would place Shivika's device there, and you would record the sound, and then that would go into your training set, and that training set would be fed into your model in order to train that model. And so your model knows the sounds based on this pre-labeled data that you already have the answers for, and then that is what you use going forward experimentally to to do stuff. But I'm sure Shivika uh, knows even more than I do about that at this
No,
point.
not at
So
all. With someone with a whole background in computer science, I highly doubt it. This is just me trying to dip my toes and a little bit into this research. But yeah, you're absolutely right, Vincent. That's exactly how we would proceed if we had human trials, I guess. We would take people who are known to not have an aneurysm, and people who do, and we record those sounds, feed it into our device model, and our device would have a little output of low risk or high risk once it's trained, obviously.
Suzanne.
This is not a question about your research, but first of all, again, mind blown. This is incredible.
Thank you.
And I can see so many, practical usage for this, not only for aneurysms, but AVMs as well, because we have different, blood flow as well.
Absolutely.
My question, so you're 17. Did you graduate from high school yet?
No, not yet.
Okay, so
I
you'll be a
just ended
senior.
school 2 days ago. I'm going to be senior. Yes.
Congratulations.
Thank
And
you.
have you thought about where you want to go to college?
Whew, okay. Well, if we're thinking aspirationally speaking with no limits or college app like process at all,
The sky is the limit at this
yes,
point.
if the sky were the limit, then it would be Brown University because I feel like I really like their open curriculum, but I'm trying to keep myself open because I know that every college has a lot to offer, no matter how big or small or how quote unquote prestigious they are. I'm just looking for a place that's going to give me really good research opportunities, let's be honest.
I was going to say you can skip the 4 years, just get your 4 year degree done next year and go right to your PhD in medical school. So pretty, no, I'm really serious because you're for someone to come up with something like this on your own without the backing of an institution is pretty incredible. So thank
Thank
you for
you.
doing this, and thank you for being so open to dive into this. It's all of us patients are really grateful. Thank you.
Thank you. Thank you so, so, so much. No, I really appreciate that. I think as much as it looks really, really cool, I do have quite a love-hate relationship with this condition. I mean, even when I'm researching it, I feel like there's still a lot of emotional baggage. So sometimes I have to just kind of take a break from it if it gets too heavy or the memories come back, or lots of things going on. But I am so glad that I was able to do something that could hopefully help all of you in the future. My grandma always taught me the importance of service, and the only purpose of my science is to serve.
Well, and also taking something that is so emotionally heavy and hard and turning that into a positive is something that takes a lot of emotional intelligence and strength, and can't wait to see where you end up. I mean, pretty amazing. Thank you again.
Thank you so much, Suzanne. I really appreciate your words. It's okay.
Shivika, do you have time for a couple more?
Absolutely, I do.
Okay, then we'll take a couple more. Anna, your hand is still up. Do you want to ask a question? Okay.
Yep, sorry, I've had some technical problems. Um, oh, I've not met you before, but
You've not?
wow,
Thank you.
I don't, I don't actually even have the words. However, so the thing I was asking was, um, so essentially we're talking almost like a Doppler situation.
Duff.
Yeah. So I understand how that works. So you're talking about the circle of Willis, which I understand is kind of near the back. Okay. So if we're talking about a Doppler that goes from here to there with your research, what confidence do you have with basically what's in between? So what research or what hypothesis do you have that would
Absolutely.
cover from
Yeah,
there to there?
yeah. Gosh, I love these questions. You guys are even better than my judges. Don't tell them that. But
Sorry.
no, it's
I'm a
great.
geek.
I
I'm a very late night geek. I'm in
No,
the UK, so
absolutely. I
sorry.
love this curiosity. This is amazing. I absolutely love discussing this and getting into this. So there is definitely going to be, like I mentioned earlier, a lot that's going to change. However, what I predict is that when we're taking into account everything in between, we've tried to kind of model that as best as we can. So oh, I forgot to mention this. The reason that we place our little mics over the eyes is because the skull is the thinnest in that place near the eyes. And it's shown that in like sound research and sound data that the sound or the attenuation, the disturbances from the skull, that's going to be minimized when you place over the eyes. And in some of our modeling, like I showed earlier, that little jelly substance that's somewhat similar to the brain because it has actually very similar density and elastic properties. So really, really great modeling there. So what I predict would be that I think that we would have a new baseline. I think that when we test in patients that we would have to just revamp our model to adjust for a new baseline reading because if we, for example, in our mathematical modeling, we showed that without all the skull and the brain tissue and the other stuff, if you just have an aneurysm vessel and you have a normal blood vessel, you would have a clearly significant difference. So even with all of the other stuff added in, I would predict that we would still have a statistically significant measurable difference. It would just be that the sound response that we get would likely be quieter, I would say. So we do have actually currently within our device circuitry little amplifiers that will amplify the signal that we get even from our models, but we would likely need to amplify our sounds further and revamp based off of the readings that we get on a healthy brain. Does that answer your question, Anna?
It does, but I have a follow-up if you don't mind.
Please,
So
please.
I'm assuming with your test data that you've had some like the audio that you've received is like a kind of like a whooshing sound,
Hmm.
like almost like the um what did I
Like
say
what brew
before?
it sounds like.
Well, like the yes, what sorry. Um like a Doppler, like I said before, sorry, like an almost like a whooshing sound. So what out of interest, just because I'm a bit of a geek,
No,
what's
please.
the sound that you get from some sort of damaged vessel? Is there
So,
some, obviously there's an interruption, but what does it
mhm.
sound like? I'm just intrigued.
So for how do I explain this? Let me pull up the original poster that we used. There's actually a really great graph on it that shows all this. If you would just give me a quick moment.
Take your time.
Where did it go? Oh, here we are. All
So
right.
I'm not trying to grill you. I'm
No,
genuinely,
please.
really,
I
really
am I
like
am
interested.
genuinely no, please. I am so excited that you're excited about this research. I'm excited about it too. All right, here we are. So I'm not going to full screen this. This was the poster I took to my science fair. But if we take a look over here, so this is all of our computational stuff, all of the fluid flow modeling that we did. And this is again, we have our CFD without an aneurysm, CFD with an aneurysm, and then we got decibel readings from that. So from that, essentially what we did was, first of all, we wanted to see, is this even significant? I mean, sometimes you have differences, but they're just noise, things like those. So we ran a lot of different I hate that word, statistical tests, essentially. And what we found was that there is a clear difference from this. If you see, it doesn't look like a big difference over here, but for an aneurysm sound responses versus normal sound responses, they're a lot more varied. So usually we see a lot more variance in the sound responses that we get. Then, of course, for our, oh, that is the model training and everything, but let's see where I can show this. For the sound that we would get, we personally, I think the sound differences are too small for the human ear to kind of detect and to distinguish that they're actually very, very different, which is why we had our machine learning model do that work for us. They're also extremely, extremely low frequencies. They're from like 0 to 10 hertz, just barely, barely audible. But our mics are actually very good at picking up those low frequency sounds. So that's essentially how we know what that so we personally, I don't really know what that sounds like, let's be honest, but our device does, based off of the data that we used for our CFD. I hope that's a sufficient answer to your question. Yes. Okay, yay. All right.
All right. We're getting two more in and we got to let this kiddo go. Let's see. Who's next? We've got Zach.
I was wondering if you could use like AI or anything like that with the research to help like speed it up or figure everything out.
Yeah. So the thing is that's actually a really, really good idea. We haven't explored that yet because especially since we were thinking about doing this kind of like as a science fair project, and also because this is our individual research, there are some ethical kind of there's some ethical nitty-gritties that we have to go into if we're thinking about using our AI for research. But especially within the research world, AI is still very questionable. Let's put it that way. The use of AI is still very, very questionable. However, there are some fields like there's AI. People are generating antibodies using AI. How crazy is that? So there are definitely some places, some spaces that AI is being used. And I would love to see if it could speed up this work because the faster we get this out, the better. We haven't explored that yet, but we would love to in the future. So thank you for giving me that idea, Zach.
All right, Jeremy.
Uh, great stuff. Two
Mhm.
questions.
Yes,
Did you do your homework before you jumped on this call?
I did. I did do my homework. Actually, no, I'm out of school, so I do not have homework.
Okay, cool. Second question, how many Raspberry Pis are you using? I'm not talking about food, everybody.
Oh, yeah, exactly. So Jeremy, so the Raspberry Pi is a little mini computer, and we use only one of them.
Okay. Between you and your partner, or you guys have one each?
So the thing is that we worked jointly together on one device, so that one device
Okay,
has one Raspberry Pi.
got it. All right, that's it. Thank
Thank
you.
you, Jeremy.
Thank you.
And Miss Stephanie, you are the last one.
Would be good if I unmuted myself. I'm sorry. Shivika, I dropped your website for NeuroAcoustic in the chat.
Oh, yeah.
And for those who haven't read our April newsletter, Shivika was featured, and you could read more about her and her amazing story and this device, her family, her work with TAAF, uh, how she's helped people learn what blood pressure is and take their blood pressure for the first time ever. So I just wanted to share that.
Thank you. Thank you so much for sharing that, Stephanie. Yeah, please
Of
feel
course.
free to. I do have a TAAF Instagram account. You can feel free to DM me there. That is perfectly okay. And I would love to get in contact with each and every single one of you. And yeah, I think the work I'm doing, it's really, really rewarding and fulfilling. I mean, helping people take their blood pressure for the first time ever. It's crazy. And it's just, yeah, so I think the newsletter gives a lot more, it's a lot more me than more of my science. But I hope that this was fun. I hope I wasn't boring, but I hope that this was really, really fun.
You did an amazing job, and from all of us, but we're going to do it anyway. Round of applause for Miss Shivika.
Thank you
Woohoo!
so much. Oh, I'm seeing the message from Anna.